Hello, this is Laura Grayson Roselli for another episode of Why Science Matters. We're going to continue our discussion on SARS coronavirus 2. And in particular, I want to talk about the vaccine today and a question that I keep hearing people ask, which is, did we skip steps to get this vaccine out to the public? And how do we do it so quickly? So we didn't skip steps. The answer to that is no. How did we do it so quickly as compared to other vaccines, which might take 5 or 10 or more years to be approved? The approval of the Pfizer, Moderna, and J&J vaccines, I just want to caveat that this is called emergency use authorization. So it's different than a full-fledged approval. And these authorizations are given when we still need an evaluation. We still need to look at whether a medication, a vaccine is safe and effective, but we try to expedite the process in an effort to get ahead of a disease. So I want to go back to December of 2019. This is the first time that we've seen this virus and people getting sick from what was then an unknown virus. And I just want to let you know that from the end of December of 2019 to January of 2020, within a 10-day period, the whole entire genome of SARS coronavirus 2 was sequenced in 10 days. Now, the human genome, which is much bigger, but still took us more than 10 years to sequence our genome. So technology in sequencing has advanced amazingly in the last several decades. And that was one of our first big jumps to getting this vaccine moving forward. Because once we know what the sequence is, and that refers to the RNA, what are the letters that make up the RNA, and how do they dictate the proteins that make up this virus? Once we knew that, then we were able to use new technology in creating a vaccine that, in the case of Pfizer and Moderna, is based on RNA. So instead of traditionally vaccines being made of the virus themselves that has been crippled, which was discussed in a previous lecture, we don't give you the whole virus. What we do is we give you the RNA. And with that RNA, your cells are able to make the spike protein. The spike protein, which is utilized by the virus to get into and hijack your cells. But that's not why we give you this, right? We give you the vaccine, the RNA, so you can make this protein so that your antibodies can be created and memory of your immune system can be stored so that when a real SARS coronavirus 2 is breathed upon you, your immune system knows how to fight it off. Okay, so the spike protein and the entire genome being sequenced was a huge advance. The idea that we are using a new format or platform also helped. And another thing that helped is in the clinical trials, this is where humans are tested. Clinical trials require a lot of people if statistically I want to know whether something works or not. And when many people are infected or afraid of being infected or influenced by this virus in some way, you're going to have more people available for the clinical trial. Secondly, we had more natural infections. So it's difficult in testing a vaccine because you're not going to give somebody a vaccine and then intentionally inject them with the virus, right? As has happened in the past with smallpox and many years ago. We don't do that now. So we had to wait for natural infections. But because natural infections were so high throughout the world, we didn't have to wait all that long for it to happen. And then, of course, there was money, right? There was a lot of government money behind it, a lot of companies and pharmaceutical industries getting involved in the vaccination getting to market. One of the last things that I'll say here in this regards has to do with manufacturing. Manufacturing happened prior to approval, which is not very common. And secondly, manufacturing was easier. Creating RNA vaccines is easier than creating viral vaccines, which is what has been done in the past. So the manufacturing, as well as the increase in natural infections and enough people to be in the clinical trials, and the advances in genome sequencing and money all came together to help us move this through. So that in February, about 1 year after the virus was first and the illness was first discovered in the world, we had a vaccine by Pfizer that was approved emergency use authorization. That's an incredible speed for it to happen, but we didn't need to skip steps. We did it smarter. Unfortunately, we had a lot of natural infections, which we don't want, but the vaccine is going to prevent that in the future. And the technology that we have been working on for decades that we're getting better and better at, we were able to bring to the table in working on this vaccine. I hope this helps to assuage any fears that you have and to answer any questions about the vaccines. Hope to see you in a future session.